so the first discussion point that we had really was to ask the panel, what do you see as being the key challenges at this intersection of AI and synthetic biology? Where are the bottlenecks? What are the main obstacles? And perhaps one way of framing that is to think about, do we need new AI, new models, or do we need new data?
Or both.
Or both, yeah.
So I'm happy to say something about new AI.
Sure.
Yeah.
Yeah.
So sure. I think we do need both. So one thing is that the existing black box AI approaches have various problems that haven't necessarily shown up in other domains, and I think these are partly to do with the fact that in science we expect explicit understandable models for the process to be carried from one generation to the next and written up in journals and so on. We, as part of that, we devise explicit hypotheses to refute. Black box models don't really support this, but I think that there's a new generation of which mixes white box and black box learning together. And by that I mean something that has both a symbolic content that tells you structurally what has been learned, and that can be read, and also takes advantage of neural net approaches in an effective way. So those approaches then allow you to build theories which can be added to, can be confirmed, can be built up over time, can end up with a kind of revised and ever revising set of theories that are being developed. And I think that's in the line, or in the best line of science, is what we should be aiming to do.
Thanks, Stephen.
What about new data? Do the panelists have an opinion on the need for new data?
Well, indeed, in response to that, actually, I was interested to hear Hector's point of view on this.
Yeah, I mean, I think it's well, you know how it is, people tend to focus on the problems that they focus on the most, right? So I think, that yes, approaches to AI are fantastic, and interpretability is fundamental. But I think before that is very much needed, we need, to create the high throughput, large amounts of data to actually leverage that, right? So I think we need both. We need new algorithms. We need an emphasis on interpretability. We need all of that, but we also need to create the large amounts of data that will make those things useful. And I think they will create the need for new AI approaches, right? So
I noticed, Hector, in the answer to one of your questions, you were intimating that, if you sort of treat it as an optimization problem, behave like an engineer and optimize, do you need that understanding?
Well, it depends what your goal is, right? I mean, often our goal, it's as metabolic engineers, is to optimize things as much as needed. However, the more fundamental understanding you get, the bigger your play space is, your phase space is to play with, right? So I think, yes, AI, we've shown we can use it to optimize systems. It'll be fantastic if we can actually develop AI to understand fundamental concepts, scientific concepts that then can be applied later on, right? That, I believe, is a future application of AI that's still being, in development.
So I would claim that understandability is as central to engineering as it is to the rest of science. So if I consider the case of designing, say, a bridge across a large river by optimization techniques, maybe it works for a while, but the problem happens at the failure point. When the bridge breaks, how do you explain it? because there'll be a team of people trying to understand how you can stop that happening again.
Sure, and I don't deny that, but if it's going to take, like 100 years to develop that fundamental understanding, there are things that we need to do right now, right? So it's going to be a mixture of both. And in terms of systems biology, we don't have maybe the 300, 500, 2,000 years of understanding of physics and material science that let us understand how to design bridges. Right now, there are companies that are trying to make products. They're going to have to deal with optimization, whereas, may take a few decades or years or maybe even centuries to build a fundamental understanding to make fully predictive models of synthetic biology and systems biology, right? Okay,
sure, but I think this will tie into the ethics issue, because if you start producing products which are dangerous to people and you can't explain why, then there's an issue.
Oh, of course, there's an issue, but the thing is, right now we had like 60 years, 70 years of molecular biology really focus on understanding the mechanisms, right? And in spite of that, we can't really predict much, right? So even though that's important, I think there are some things you can do right now with optimization and that are going to be useful and can be proven to be safe beyond the fundamental understanding that you're seeking, right? Well, at the same time, we seek that fundamental understanding that may take a significant amount of time to get, right?
Mary, I think you had your hand up earlier.
Yeah, I was going to jump in, like something from the synthetic biology perspective that would be useful for my lab, and I think many other synthetic biology labs, is generative models, and this is something that's starting to come into the space. But people have been working on synthetic biology parts like ribosome binding sites or promoters, and generating data there, and then using them to develop different machine learning models. And you can learn a lot about the components that go into what makes a promoter really effective as part of that. But at the end of the day, it'd be really nice to have a tool that could actually just generate a strong promoter sequence for you, or a medium promoter sequence. And there are tools out there that can do this, but I think this is a really exciting and emerging space. And in particular, there are a lot of sort of nuances. Like if you want to design one promoter, that's one thing. But if you want to design many suites of them, then perhaps you need them to also, not just like the sequences need to be different enough. And so they have to be dissimilar in addition to being, effective at their job. And so you're optimizing for a number of different things. And I think we've seen tools that offer potential promise in synthetic biology, like the, ribosome binding site calculator, that are based on thermodynamic models. I'd love to see really powerful tools that come out of the AI space that could generate synthetic biology components that would be really useful. Yeah.
Eduardo.
Yes, I wanted to add on the topic of interpretability of the AI models. I do think that, having explainable AI models many times can be helpful for situations involving some degree of accountability, maybe life or death decisions, or if you're, building a bridge that might fall, or if you're like programming a self-driving car that needs to change lanes, or it may be helpful in other cases where you care about the fairness or the visibility of the outcome. But for many industrial applications, like Hector was pointing out, if you're optimizing, let's say, as I was mentioning in my talk, for the manufacturability of a specific compound, I would tend to believe that you care less about the interpretability, and as long as the algorithm can help you design a better strain that can, achieve a higher titer of a metabolite or a molecule. However, I do agree that many times, when working with biologists that are looking at the outcomes of the models that we train, they want to understand the algorithms and why the algorithm picked a certain set of inputs. And many times they are surprised by the outputs of the models. And sometimes it has to do not with the model, with the algorithm itself. Many times, the quality of the model was affected by the training set used for training the model. And that's when sometimes it's kind of good to be able to explain the models. Also, of course, when you're doing basic science, you want to understand how the biology works, and or maybe when you're moving to engineering human cell lines, humans really want to understand the model before taking any actions that might have some unexpected side effects.
Actually, Hector, that reminded me of something I was thinking during your talk. It's a little bit more of a technical question, but you seem to do something slightly different to others. And you seem to have a variety of models, a kind of random forest and and then you were sort of ranking those models, or were you?
Yeah, well, based on how good they are, based on how good they are, they're given more or less weight, right? In the final approach, right?
So you're then just sort of using kind of an amalgam of different models in terms of what kind of maps to the output.
Yeah, it's an ensemble model, and based on how successful they are, they're given more or less weight, with in a Bayesian inference way. Yeah.
Okay.
But I think
I might interject a question from the audience here, which comes in quite handy if we are talking about working at the intersection of AI and biology. How should a biology background PhD student pick up AI and integrate AI in future research?
Can I jump in on this one?
Yeah.
Sorry. So I have a couple answers to this. So one is that I'm on sabbatical this year, as some of you know, and part of what I'm doing is sort of diving into deep learning literature, and so this question is very near and dear to my heart. I should say that there are some really phenomenal resources out there in terms of online coursework and tutorials. That it's not SynBio specific, but there's some really good material out there. Also, universities presumably offer courses as well, but I think that you can do pretty well. There's a lot of information in this space. The other thing I want to mention specific to SynBio is that I'm working through an effort with EBRC, the Engineering Biology Research Consortium, to make a education module on synthetic biology and machine learning, where we're talking with a number of people in this space, and so that should be available on the EBRC site in early summer. So that'll be something specific for SynBio that may be useful to some people here.
So yeah, keep tuned and keep an eye out for these. Let us know, Mary, when these are out. I see another interesting question relating to what we were asking about data. Do we need the new data? Colleague Spencer is asking us, if you had unlimited resources to generate a data set for synthetic biology, what would it be and why?
Yeah, maybe I could jump on this. So yeah, with new data and new AI. So new data. So what you have to be careful with data is not because very often we say we have a large amount of data. Okay, we can train. Let's say, for instance, omics data. So, but when you do engineering, what matters is not that you have a lot of omics data on one engineering example. You'd rather have many engineering examples, okay, than a lot of data on one example, okay. And because what you do when you have omics data on 1,000 data points, you just increase the feature vector that you're feeding into your system. So that's very important to have many examples. So one way I would say if you have a fixed budget, okay, one way of reducing the amount of data that you need is to basically bridge these mechanistic models. So if you're dealing, with a strain model, to bridge these mechanistic models with this AI technique. This is called hybrid model, by the way. And also sometimes scientific machine learning. And this is what people have been starting to look at. Okay, so for instance, they have merged ODEs, within the scientific machine learning community. They have merged ODEs, with machine learning, for instance, where you could find out parameters that you are lacking, for your. So I would say new AI, yes, hybrid models, and the reason would be to reduce the amount of data that you would need, to solve your problem.
I might also add one point here. I think that we still lack, contrary to other fields where we use extensive data sets to do model prediction and to infer what the future might look like. In biology, we still lack the capability of tracking multiple molecular species or molecules, if you want, over time in a non-invasive way. So SynBio has two key modalities to collect data, right? You collect data from millions or billions of cells, but at one time point. That's what you would do, for example, with flow cytometry. Or you collect data of one or two, or maybe, if you extend and you push really hard, maybe 10 different molecules over time, but not more. And even when you do that, typically you don't do that at a single cell level, you do that at a population level. With microfluidics, you might do that at a single cell level, but it becomes tricky. So if you think about the cell as being the system you want to design, with all of its aspects, stochastic gene expression processes and everything that it encompasses, we still are not able to have more than 10 maximum, big maximum, 10 signals that we can track over time in a non-invasive way in a living cell. So when you ask for, if we had enough resource, what would we measure? Well, I would pay quite a lot to be able to measure, 1,000 molecules, for example, simultaneously, non-invasively at a single cell level.
The challenge to me, the challenge to me appears to be how to reduce the amount of data that you need, and there are fundamental laws that are central to machine learning, and one of these is the no free lunch theorem, which says that basically you can't learn if you don't have a bias to your learning. So if you have completely flat priors, you can't learn anything, and the more bias that you have, the less data you need. Okay, so that's the data efficient approach, which actually seems to me the preferred engineering route. Normally, you're not trying to maximize the amount of resources that you have to use, you're trying to minimize it. To do that, you need stronger biases, and that means that you should have background knowledge which is built up incrementally. I think that's the only way that we know of doing that very effectively. Unless you're going to have to engineer every bit of the background knowledge, you should use the machine learning to help you build the bias up.
Sure, but there is a recurrent argument here, right? To build the bias up, to build the background knowledge, you need to also, you probably need to extract or perturb your system in an informative way, so therefore extract data.
Yes, you have to carry out experiments like a child does in the world. And you start with very little, but as you build it up, your ability to learn becomes greater and greater. It doesn't stay the same.
Yeah. I wish everything is encoded in an experiment so that we know exactly how the data or how the information is extracted from the system, which in biology seems still to not be the case.
There's a nice question from Robert Endres asking, is AI a substitute for or a shortcut to understanding? So like the concept of systems biology or physics informed neural networks, as they provide natural science constraints. Natural science constraints for mechanistic insight. So is AI a substitute or a shortcut to mechanistic understanding?
I hope it's a shortcut. It depends how you use it.
I think, it can be a shortcut, right? And sometimes you may not need more than the shortcut, right? I mean, look, there's plenty of companies out there who are serving us ads, every day, and they have no interest whatsoever, in the fundamentals of our brain. They just use machine learning to do that. However, I think the challenge here is how we can use AI to really generate understanding from a fundamental point of view that we can share with other people, and being able to have AI, like in the case of the survival lab or the automated scientist, how can it really develop the next set of experiments to create fundamental knowledge that it can be used as biases, as priors, as you want, to really not just solve the problem that we have at hand, for this pathway and this product, for example, but create fundamental understanding that they can be expanded, to solve other problems later on. I think that's the key challenge there, which experiments and which techniques to use that.
Yeah, the techniques have to come from transfer learning if you're going to transfer what you've learned to other tasks.
Well, that's one solution, yes.
Yeah, or like we talked about earlier, is that this merging with having a mechanistic layer, within the AI process. where you can actually interpret, the weights like they have done in signaling networks.
Sure, but that needs a human to actually put the mechanistic layer. Can we actually have the algorithm create a mechanistic layer? I think that's the challenge.
Yes, yes, you can, yeah. Yes, you can. You just have to have, if, for instance, if you work with a neural network, rather than having the classical topology of the neural network, you replace that in the signaling network. That's what they did. You replace that by the topology of your network. Then sure, the weight become meaningful. Then the weight become meaningful.
Sure, but that topology of the network was put by a human there, right?
Was that?
It was a human that put the topology of that network.
Yeah, sure. Yeah.
Infer that topology automatically. I think that's.
So what if there's errors in that topology, Jean-Loup?
Yeah, well, yeah, but these have been worked out, for many years. So these topology.
So I think earlier in the chat, I think Nathan Hilson had an interesting question. He was sort of saying, could you have theoretically sort of, 1,000 different variants of your model and sort of evaluate which one is best? Can you use sort of pathway discovery?
Yeah, well, you would have to have changing topology as you go. Okay, so maybe this could work. This is not something I thought of. Okay, how to actually work this out. Okay, but you could think from the weight maybe to knock down some. Definitely, what you can do with a fixed topology when you start from, you can easily knock down some paths, some connections, meaning pathways, okay, or reactions. And you can definitely do this. You can also add some, yeah, I guess, and see how that would behave, But one thing at a time, okay. For the mechanistic, for the metabolic models, the first thing was to get the intake rates. The second thing to look at is the biomass reaction coefficient, okay, which are kind of weird, okay. And there are other few things like that can be worked out. So this way you do change your model, But now changing the connectivity of the model, it's another thing. But for metabolism, reactions have been worked out for years. Generally, we have a good idea. If the enzyme has been found in the strain, you have the CC numbers, you have the reactions. So there's a lot of work. But 50 years.
So yeah, I mean, I agree.
So for that, now signaling network and regulatory network, it's more dicey, okay? Because granted, these are inferences made on whatever technique they use, So it's not as clean as a metabolic network, but the metabolic network has been worked out for some time now.
I agree that we have a lot of knowledge of metabolic network, but I suppose flux balance analysis doesn't have a great reputation.
Yeah, but the stoichiometric matrix, so that is the network itself is correct, I think, okay? Or maybe few things need to be added or removed, But what's missing is the intake fluxes and the biomass objective function. Okay, so and few other things like that. There are some few other things that need to be fixed. So this is where machine learning can help, in order to find this out. And so similarly, people also have been looking at that with ODEs, on how to solve and to tune the parameters of set of ODEs, in order to study the dynamics of not necessarily a pathway. It can be many things, And this is really happening now. So it's something the SynBio community should look at, I think.
So one further point, I wanted to give the panel one word: standards.
Yeah,
the standards are needed in SynBio. Okay, that's my take. Okay, once we have clean standard in SynBio, the AI could pick that up. I don't know, maybe I'm wrong, but I mean,
we're all making our life much harder than it needs to be, right? By kind of like not adopting standards. I mean, and what standards? I mean, I guess the reason we don't is because we don't have to publish, right? We can just publish relative fluorescent data, and no one really cares. But data comparability and data transferability, I think, is kind of probably hugely kind of holding us all back here in terms of our ability to integrate across the effort that we're all going to.
Yeah, the very minimum surely is that you need open source availability of the data, the models, and the systems that are being used. Surely. And because even without that, you can't even reproduce the results that have been published.
Yeah, I don't think anyone disagrees with that, but I've been to many panels like this, and we always say data is important, data standards are important, we need to be able to exchange data, and no one really does a lot about it, right? I mean, some people do things, they try, but it's not the kind of things that has the high priority funding that it would deserve, right? I guess that's the question. When does that happen? Everyone says it's important, but we can't really convince the funding agencies to fund it like it deserves. Why is that?
Because the publishing houses are more interested in the latest discovery than in than in funding standards, right?
No, but the publishers could demand that certain standards are met when you submit a paper, or else it's not accepted. And making all of your stuff available is a start on that. And if there is a standard, then saying that it has to be according to that standard.
Yeah, there are some, like Guy-Bart, but it's true, we are not mandated to.
I think there are some developments here. So, you know, there are now sort of minimum information standards for, you know, things like kind of qPCRs and stuff like that, because they kind of realized that the stuff that was being published was, you know, not necessarily well founded. But I do think that, you know, there is a really important question there around, you know, the incentive structures of funders and publishers, which, you know, and also us as researchers, right? You know, we're not incentivized to kind of work in this field because it's not funded properly, and it won't give you high profile publications either. So I think it's a really difficult problem to address, and also kind of coming up with standards and getting people to adopt them is a really thankless task as well. But I think everyone kind of like really agrees that it's kind of really important, but I just think it's super challenging as to how we can actually move that forward.
Yeah, I would say that, you know, maybe we as a company, we are a little bit more, you know, motivated to, you know, help develop some standards. You know, we've been using and adopting a number of, you know, standards already, you know, like such as SFOL and others. But as I mentioned in previously in my talk, we've been working on looking and thinking about standards to represent also, you know, not only designs, but also experimental protocols and workflows. And I do think that I tend to believe that standards will become increasingly more important, and there will be kind of like an increasing need to collaborate more. Like it used to be, you know, a long time ago that, you know, a lot of, you know, a lot of research and discoveries could be done, you know, on by individual contributors, but, you know, the challenges that we have ahead will demand collaboration not only between academic partners, but also many times between companies. We see the need of companies, you know, collaborating with academia and with other companies. So we're kind of like helping develop some standards already in collaboration, for example, with Biomed, the Biomed Institute, where there is a group of 100 institutions, half of them companies and big corporations, and the other half different academic universities. To specifically, you know, this one is for applications on optimizing fermentation and some other industrial biotech applications. Yeah, I agree. It's like many times something that nobody individually wants to do, but they are important, and sometimes they require, you know, the push of maybe a government agency to have them being developed.
So I was interested with, you know, your software platform, Eduardo, and also Hector, your EDD platform, which is a data platform. You know, are standards, you know, do we need these kind of platforms to kind of really help bring standards on? That, you know, can we begin to incorporate standards into those platforms so the data kind of semi-automatically gets
Yeah, we are working on that, right? So we created ADD, and then anyone could put their own copy of the ADD, and people did. But then things, the data and the databases in each of these started, drifting away from each other. So there wasn't a way to share things. Now we're working in a way that we could actually share data between the ADDs, and hopefully that can be a standard for sharing at least omics data and phenotyping data, right? So we are working on that. But again, when we talk about these things, everyone is interested. It's really hard to find the money to actually pay for it. And software engineers and the data scientists that develop these ontologies and these standards are really not that cheap, and they are hard to compete by in the Bay Area, right? So those are the issues that we have with that. If we could get it funded, I'd love to, to work on that. And we have found people who are really interested in doing it, but it's really hard to find the funding to do a really good job. You can do an okay job, but to really do a good job is hard to find the funding, in my experience, at least.
Yeah, I agree.
And so in the electronics industry, people depend on ISO standards, the international standards organizations. So why doesn't the community set up an ISO standard committee?
There has been quite a lot of effort actually to interact with the National Institute for Standards in the UK, in the US, and in other countries too. And they've tried to set up these things, but I think there is the issue that we have been talking about in terms of the funding, and also the reluctance of some of the users to become confined and constrained into a particular standard as well that has come forth. So it seems to be a difficult problem to solve. We have been trying to solve it for more than a decade. And still we are battling with it.
And I think those sort of ISO standards are very framed around industry needs. And I think still a lot of, the kind of things that we're collectively talking about are probably somewhat upstream of industry. I mean, Eduardo, you've probably got a bit better insight here into, what point do the sort of standards for, the kind of synthetic biology manipulations and measurements that we do kind of impacting in industry. And I think, downstream, once you kind of get into the manufacturing domain, then there's much more rationale for having standards, particularly if you're sort of looking at kind of pharma kind of end products, because of the regulatory framework. So
yeah, no, you're right. That's exactly what we've been kind of now looking into, how we can help define standards for defining and executing experimental workflows that come downstream the process of designing DNA or synthetic biology systems. We've been looking, for example, into PAML, the protocol activity modeling language, as a way to represent and exchange protocols between labs, in particular SynBio protocols. I mentioned that we're working with Biomate in the context of industrial biotech applications, but yeah, those standards, the ones that we're looking at, should contain for the cases that we need to cover for industrial applications. They need to cover a good representation of an experimental protocol, and also many times specifications about the execution context, around running a protocol, like the equipment that was used and the results that were generated, and the specifications about the protocol run, like when was it run, by whom, and so on. And those are things that are of less interest to maybe academic researchers, but are needed in companies when they need to start scaling or collaborating with when they start expanding and they want to open a new lab somewhere else, and they need to replicate and continue doing experiments in different contexts. There needs to develop standards around those downstream processes, as you were mentioning.
Mary, you had your hand up?
Yeah, I was just going to throw in a slightly different perspective, which might be interesting here, which is one of the things that we found in doing image analysis is that, we've generated a bunch of microscopy data on our lab, microscope, and then we included that in the training set. And we, have 2 microscopes, so we've got 2 microscopes worth of imaging data. But then once we started getting data from other labs, they didn't really use exactly the same protocols, and their cells look a little bit different, but they were able to use the imaging platforms the algorithms we developed, and then we can feed that actually back into the training set. And as we gather more data from diverse labs, the model actually gets a lot better at generalizing to different conditions. And so I think this is kind of an interesting perspective, and I'm not entirely sure how this generalizes to sequence space or other things that might be relevant for synthetic biology. But I think one role that the training set could play is that if you have diverse sources of data, that might actually allow you to capture some of the diversity you see in terms of how it's generated. So it certainly works at the imaging side, and it'd be fun to see how it extends.
Thank you. In the remaining time, I wanted to jump into a different direction for the conversation. So obviously, the confluence of AI and synthetic biology brings together the 2 technologies with the greatest transformative potential for our world, at least arguably. Is there anything we should be worried about? And maybe I would invite Claire to give us a little bit of a perspective on this.
I knew that was coming at me then. So yeah, I guess there's obviously the problem of it being used not in the way that it's intended, which is the typical problem with synthetic biology is that of nefarious use, as well as that of public perception. But also I think that we've already discussed that a big problem with this or with AI automation is that it requires such dependence on an initial data set that data set has to be rigorous. And there's problems with ensuring the integrity of a data set, both in, its initial, content, but also in the biases and whether the biases that you're putting into it are the correct biases. Yeah. So, yeah, that's me.
Yeah. I mean, the question of bias in your data and the difference that exists in data from natural systems versus engineered systems and design systems. How can we truly predict what could happen with inserting new things into a given organism?
Yeah, and I think going back to what Mary said about how it worked within the imaging system for if you were to give a separate protocol to different labs, then you could still use that data then. But having worked in a field where we used a metabolic model to simulate human digestion, it was the same protocol used for every lab, but the results were not consistent depending on, it was so subject to, even the temperature within the lab, whether the timings of which you did it, what your interpretation of after 30 seconds, for example, was, that it wouldn't have been usable as a data set. Whether you put it in after 30 seconds or on 30 seconds, I feel like if you have a data set, I think the problem can be that you can assume that your data set is suitable, but in real terms it isn't. And also,
these points sort of feed back a little bit into the data standards, right? But in metadata standards. So, actually what descriptors of the experiment do we need to capture alongside the actual experimental data?
Yeah. But I also think that maybe it limits the possibility for real innovation, because you're basically having your AI produce predictions based on set patterns that you have. But what if you're missing something completely separate from what you've even considered?
I guess that sort of relates a little bit to the, scientific discovery, right? Which kind of stems from the unexpected observation that someone has the gumption to follow up. and to nail down and actually figure out that there's something really interesting going on there.
Yeah,
entirely unexpected.
But I think this is the problem, because my background is in classical biochemistry, and then moving to synthetic biology, that already was a bit of a leap. And, you know, so I think it does depend on which mindset you're going into it as well.
And yet also complementary to what you say, methods like machine learning and AI could actually find some patterns in the sea of data that we already have that might not be that trivial or intuitive to see by the human eye. So there is a counterpart to it as well, where this enabling technologies allow us to actually find information in the data that are not necessarily that easy to extract, at least for a human being.
The question about biases in the data is related, I think, to the black box issues that I was talking about as well, because if the model that will be learned will reflect the biases of the data. And you might, if you used a black box data system, simply accept the model as it is because you don't know what it says. In the case that it actually reflects a contentful description of the data, it'll be much clearer what it was biased about if you can recognize the pattern, if you can recognize what's being said in the model. It's much easier to debug the data if you've got a program rather than just a black box.
Yeah. Any other aspect that the panelists think we need to be cautious about, careful about when we work at this confluence of AI and engineering biology? Would we trust our designs?
I suppose for me, we always have to be mindful of what it is we're trying to achieve, and I think for many of us who are sort of working with microbes trying to do fairly fundamental things, it never feels particularly ethically challenging. But nonetheless, I think in terms of I suppose one of the things that does concern me is I feel that there's an awful lot of potential to contribute positive change to society, but whether society wants to accept that change. We've seen in Europe huge pushback against GM crops in the 1980s, which is a position that's still extant, right? It's still extremely hard to grow and develop GM crops in the EU, and that for me is kind of still a real concern, that we can put all of this effort in and it won't lead to the kind of positive impact on society that I think everyone here wants to see, right? We can see that the problems arising from having a fossil fuel based economy, and really that transition to a bio-based economy is a huge opportunity, but one that needs to be managed. And I don't really know quite how we manage that.
I agree. I think, some level of regulation is needed, but I'm also sometimes worried about unnecessary regulation. Regulation is needed. Like I'm not concerned about the scientific community, but there's some concern today around the development of bioweapons, and I think that's a real threat. And the weapons of the last century have been physical and chemical, and those of the 21st century could be easily biological. But so there's regulation that is regulating companies synthesizing DNA and so on to screen what is it being synthesized by. But on the flip side, I think we've also seen sometimes regulation that might slow down development. That we have to carefully balance how much we need and or not.
Yeah, I mean, I think so. I mean, again, in the, there are regional differences here. So, I mean, certainly in the UK, there's really pretty strong regulation around any kind of GM modification of microbes. E. coli and yeast, even the standard sort of workhorses, and so I think vast amounts of synthetic biology, certainly in the UK, are covered by existing pretty comprehensive safety and governance, which I think is appropriate, but that's not necessarily the same kind of universally across the world. I mean, I think it is much lower barriers to entry into that in the US, and you can argue whether that's a good or a bad thing. But I suppose there is also always that question of is it appropriate that certain work should be done? There are sort of questions around COVID-19 as to whether did that arise naturally or not, and I don't think we will know the answer to that, to be honest. And that's really troubling, and that should we be doing forward evolution of viruses? And I think that these are really valid, important questions that I think our regulators need to get on top of, because if they don't, then it kind of deprecates everything else as well.
Yeah, no, I completely agree. Like, for example, take coronavirus, that caused COVID. It's a very small genome, like 30,000 nucleotides, encoding, I think, roughly 30 genes. And, the cost of synthesizing that genome today is, quite low. And these genomes can be built today and optimized with our current technology. So we have to be paying attention.
There are ethical considerations about genetic modification, but there are also ethical considerations about the development of AI. And what we're talking about is a field that has to inherit both of those discussions. So I don't know how they interact, but maybe if we have more discussions like this, we'll understand that better.
Yeah, that's a good point. Yeah,
indeed.
Yeah, I agree. I think it's a perfect point actually to end up the session. And I would like to take the opportunity to thank all of the speakers and the panel members for the input, and the audience for staying with us until the end. I hope that this has stimulated your reflection around work at the confluence of AI and engineering biology. I invite you to continue the conversations and to get in touch with the panelists and the speakers to continue these conversations. I'm sure that we will see more coming at the confluence of these two fields. And I think that we also have the responsibility to discuss and develop this intersection with these key questions in mind of responsible research and innovation when we do such work. So thank you all again. It's getting late here in London. I wish you a very good morning, evening, afternoon, depending on where you are in the world, and I hope that we will have the opportunity to take this up to the next level in the follow-up of this event.